The G-protein coupled receptor is a plasma membrane receptor which works with the help of a protein that binds to energy-rich molecule to the energy-rich molecule GTP called the G-protein. G-protein coupled receptors are used by various signaling molecules such as yeast-mating factors, epinephrine, and other hormones. The binding sites for the ligand and different G-proteins inside the cell vary for these receptors. The structure of G-protein coupled receptors consists of 7 alpha helices spanning the plasma membrane. Specific loops between the helices form binding sites for signaling in G-protein molecules. These receptor systems are crucial for both vision and smell in humans. The similarities among the structure of G-proteins and G-protein coupled receptors in diverse organisms suggest that these evolved early.
The G-protein is attached to the cytoplasmic side of the plasma membrane and acts as a molecular on-off switch. The on-off position depends on the presence of a certain nucleotide, either GDP or GTP. When GDP is bound to the G-protein, it is inactive. The receptor and a protein work together with another protein, most likely an enzyme. Go.
A specific signaling molecule binds to the receptor on the extracellular side, which is this part. Then the receptor activates and changes shape. Next, the receptor binds to an inactive G protein on the cytoplasmic side. This causes a GTP to release GDP. GTP releases GDP, which activates the G protein.
Once it is activated, the G-protein leaves the receptor, diffusing along the membrane. It will then bind to some enzyme, altering the shape of the enzyme as well as its activity. The GDP on the G-protein activates the enzyme, allowing it to trigger the next step in a pathway leading to a cellular response.
After the G-protein reacts with the enzyme, a phosphate is released from the GTP to make GDP again. This is done by GDPase, an enzyme which is a function of the G-protein. The enzyme is now inactive again, and the G-protein is able to be used again. The GDPase function of the G-protein allows the pathway to shut down rapidly when the signaling molecule is no longer present.